Good morning, everyone. I'm Brian McCourt, president of the McCourt Foundation, and we'd like to welcome you all here today for the first ever virtual McCourt Foundation educational update. We've got a fantastic lineup of physicians that we'll be presenting today. Physicians that are not only top in the country, but top in the world in their respective disciplines. We're excited about having you all here and hosting you, and would also like to thank all of our sponsors that are making this possible. But I'd like to especially thank our presenting sponsors, Bristol Myers Squibb and UBS Financial, as well as our silver sponsor, Sanofi Genzyme. But I would like to thank all of you for being a part of this day. This has always been one of our favorite and most impactful programs, and it's all because of you. So enjoy the program. I'd like to now introduce Steven Sypek with UBS Financial.
Thank you, Brian, and thank you for joining us today, and welcome to the McCourt Foundation Educational Symposium. My name is Steve Sypek, a financial advisor with UBS Financial Services here in Rockland, Massachusetts. I've been friends with the McCourt family for many years and personally involved with the foundation for over 20 years. I too have been impacted by neurological disease, and that my mother passed away from Parkinson's 5 years ago this year. I am excited for today that you will be hearing from the top doctors and researchers from Massachusetts General Hospital, Brigham and Women's Hospital, and Cedars-Sinai Medical Center on their progress and developments in the areas of multiple sclerosis, Alzheimer's, and ALS research. We all know the challenge that individuals and their families face in dealing with neurological diseases. And that is why I believe the McCourt Foundation focus on making things better by supporting research, providing education, and developing strategic health-focused partnerships will one day lead to a cure for many of these diseases. As a financial advisor, I know the challenges that these patients and their families face in planning for the unexpected and in dealing with the emotional, physical, and financial challenges. We believe that the financial services industry can help families plan for those unexpected circumstances, and also help support the critical research and awareness in the areas of neurological diseases. And that is why UBS is excited and proud to support the McCourt Foundation in their great work and their mission by sponsoring today's educational update. Thank you for joining us. I hope you find these sessions helpful, and that they will continue to inspire and empower you and your loved ones in making things better each and every day. Enjoy the upcoming update.
Good morning. This is Dennis Selko, reaching you from Boston, and I'm delighted to be here with my colleague Mark Albers. We are both neurologists working on the forefront of dementia and Alzheimer's disease and related disorders. And this morning, through the McCourt Foundation, we have the pleasure of telling you about a little bit about the disease, answering your questions, and about our work on the disease. So with that, I'm going to share my screen. Let's see if I can find it. Here it is. All right. I'm hoping that you're able to see this now. And what I thought I would do, and Mark will do this similarly a little in just a few minutes, is to give you my sense of some of the issues we face in dealing with this tragic disorder of Alzheimer's disease, which is so very common. And we'll share with you some of the knowledge that the field has acquired and some of the things we still don't understand, and a little bit, most importantly, about what I'm sure all of you care about, which is the therapeutic approaches to this disease. And both Mark and I will speak to that. Uh, so let's talk a little bit about where we stand and where we need to go. Alzheimer's disease in 2021 is the most common cause of dementia, and I'll define some of my terms in a moment. It affects well over 5 million Americans, probably 40 to 50 million worldwide. We don't know the exact number. And that number probably is an underestimate because the field has redefined Alzheimer's in the last decade or so as the disorder that causes abnormalities in the brain, and gradually those abnormalities build up even prior to the first symptoms, the first time you forget where you parked your car or forget the name of your 10th grandchild, if you're so blessed. Those early symptoms are preceded by a long period of abnormality in the brain. So in some sense, there are any number of millions of people incubating the disorder who aren't aware yet that they have symptoms. And this is true for many chronic disorders like hardening of the arteries, atherosclerosis. It's there for a long time before you have your first twinge of angina. So Alzheimer's occurs in all races and ethnic groups across the globe. It's relatively similar in its prevalence. And people in America of African, European, Asian backgrounds will all experience Alzheimer's, and it'll be a prevalent disease in their society. We see it in China, in India, Japan, all over Europe, and so it's a universal foe of good health. It's expensive, and here I quote a figure of about $250 billion in annual cost. We, again, don't know the precise number. It's probably north of that. And it recently was estimated in the New England Journal of Medicine that the cost for all aspects of Alzheimer's, principally for caring for patients during the long time that they suffer the symptoms and the neurodegeneration of the disease, that total cost for our society may be more than the combined cost of all of hardening of the arteries, that is atherosclerotic disease, and all of cancer combined. So all of cerebrovascular and cardiovascular disease plus cancer, the cost of Alzheimer's for our society is still greater. And yet the amount we spend on research is not in proportion to that. And I'll mention that there's no cure for disease-slowing treatment at present, but both Mark and I will speak to how we're hoping that's going to change, perhaps even relatively soon. It's an exciting year where we're going to talk more about therapeutic opportunities. So to share with you some of the current concepts, senile dementia is a term we use in medicine. Senility is a lay term, and we think of it as progressive mental failure after age 65. So this is the general formal definition. We know there's no strict cutoff, and there are so-called pre-senile forms before age 65, and that's really a continuum. So it's not like two groups of people. The disease can occur rarely in even the 30s and 40s, that's very unusual, and then in the 50s and 60s, and increasingly more prevalent as people get older. Alzheimer's is the most common of maybe 20 causes of this concept of dementia or loss of mental ability that comes with age. And it may account for 60 or 70% of dementia cases in the US. But I should say that many of those 2/3 of dementia cases that have Alzheimer's don't just have Alzheimer's. They may have other changes in the brain. Commonly, they may have minor or major vascular abnormalities, microvascular disease. And that, of course, makes it harder for us to devise treatments for Alzheimer's because the disease is not entirely pure. It's more pure in people who have hereditary forms that begin in midlife. As you read here, Alzheimer's begins insidiously, usually with decreased memory, confusion, disorientation, maybe some word-finding difficulty, and it progresses slowly over 5 to 20 years. It is fatal. People are sometimes surprised at that, but many neurological diseases are ultimately fatal. They don't directly stop the heart from beating or the lungs from expanding, but they cause so much debility so much difficulty that patients have a variety of secondary and tertiary complications. And many of my patients with Alzheimer's will pass away at the night when, even as good as their care may be, there's a time for sleep for both them and the caregivers, and they will have a little bit of aspiration or some phlegm getting stuck in their throat, and that final respiratory event is what will take their lives. But the reason they died, of course, was because of this progressive Alzheimer's. Here you see a list of other types of late life dementia, and Alzheimer's disease is at the top of the list because it is the most common we see. It used to be that vascular dementia, or what has been called multi-infarct dementia, multiple small or medium-sized strokes, could be considered the most common cause of dementia. But with the 50% improvement in atherosclerotic disease and hardening of the arteries in our society, there's less of that now. And we've controlled blood pressure, to some extent diabetes and cholesterol levels pretty effectively. So we see somewhat less of that, but it still happens. And some of it happens in a way that the patient and the doctor can't tell. Parkinson's disease with dementia is also common. Parkinson's in general is about a third or a quarter as common as Alzheimer's as a disorder. And most people with Parkinson's eventually will develop dementia. A special form of dementia we refer to as dementia with Lewy bodies, and that's related in part to Parkinson's disease with dementia. But when the Parkinson's changes and the dementia begin essentially at the same time, then we think of dementia with Lewy bodies. Whereas in Parkinson's disease, you begin with the features you may know, with trouble getting out of a chair, walking, tremor. They come first, and the dementia comes years later. But dementia with Lewy bodies, both dementia and a Parkinson-like picture, arise simultaneously, and it's a very difficult disease. Frontotemporal dementias are another large category, including Pick's disease and other diseases that have unusual names. And these are sometimes due to the protein tau that I'll mention. And then Creutzfeldt-Jakob disease and other diseases that are so-called prion disorders are very rare in society. One renowned and notorious example was what's called mad cow disease that occurred in Britain 20 years ago or so, and can still happen, but that was a very unusual situation where an unusual form of infectious particle called a prion was in the brain. That has really very little to do with the other disorders. And so Alzheimer's disease is at the top of the list. But we think about this differential diagnosis when we see patients in the clinic. Our job is to figure out what form of dementia, and there are others not listed here that could be causing the patient's symptoms. I want to mention here, and this will be relevant to Mark's work and his presentation, that we generally separate treatments for any disease as symptomatic treatments versus disease-modifying treatments. So for Alzheimer's, the symptomatic treatment are the so-called acetylcholinesterase inhibitors, and they have names like donepezil and razodine and galantamine, and these are drugs that have been on the market for 20 years. The most well-known brand name is probably Aricept, but there are others, Exelon, razodine. And so these are agents that help the symptoms for a little while. They help the loss of acetylcholine in the brain, but they don't really slow down the course of the disease. And that's been looked at carefully, and there's very little evidence that they slow down the course. They may be helpful for a few months or a year, or sometimes a little longer, but they're symptomatic. They're not disease modifying. What everybody cares about is disease modifying treatments, and we'll talk about those a little bit more. To get to disease modifying treatments where you actually, for example, an example of these two classes of drugs would be someone who comes forward with a sinus headache, and I tell them, well, you have a bad sinus infection. Here's some Tylenol. You're going to feel better tonight. And they look at me puzzled and say, doctor, I didn't need to come to see you to get Tylenol. And then I'll say, well, of course, we need to give you a prescription for penicillin. And in Alzheimer's, we just say the first, but we don't have the second. We don't have the treatment yet to offer that really slows the disease. So to get to disease-modifying treatments, we have to really decipher the disease. And here you see a picture of a part of the brain that is important for emotional control called the amygdala, and behavioral symptoms can arise in our patients when this area is affected. Also, the area called the hippocampus and then the cerebral cortex will have the abnormalities you see here. So in this image, which is a section of the brain, the area that, for example, is in the upper left or the lower right of this slide is how a normal brain might look. There would be no large round lesions like you see here, and none of these very dark brown nerve cells or neurons. So the lower right area would be the normal situation, but this patient has, in this high power view, 2 amyloid plaques. So the amyloid is the central material, and it's made up of a protein called A beta. And the title of my slide mentions amyloid plaques made of A beta. And the plaque is surrounded by or permeated with abnormal axons and dendrites. These are the nerve processes that connect one nerve cell to another. Sometimes they're very dilated, like you see here. And so if you are medically connected and you're attending this talk, these are so-called neuritic plaques. They have abnormal axons and dendrites or neurite. Adjacent to the 2 plaques in this patient's brain are tangles. These are so-called neurofibrillary tangles made up of a protein called tau. And the cells with this particular stain of the brain should look sort of like this, and not with this dark brown material that's sort of stuffing the neuron. This happens to be a brain section taken from a patient who I followed during life, and he died at age 69. And his mother had Alzheimer's before him. And we didn't know it at the time, but probably he may have carried the so-called ApoE4 allele that we'll talk about in a moment. So these are the plaques and the tangles. The plaques are the round spherical lesions, and the tangles are inside the neurons that are the hallmark of the disease. And that for decades, people like Mark and I and people before us have been trying to decipher to understand, are they the cause of the disease, so to speak, and can we interrupt them? Now, I'm going to give you an example in this cartoon I'm going to show you of how we think about the amyloid part, that is the plaque part of the disease. We think about the amyloid protein in these plaques arising from a cutting process. Here is a cell, a cartoon cell with a yellow cell wall. It could be a neuron. Actually, this could happen in muscle cells and skin cells and elsewhere, but in the brain, we care about neurons. And in this neuron, there is a protein in this simple cartoon in red and black that's called the amyloid parent protein, or APP. And I'm going to show you that the generation of this amyloid beta protein, which is the black box here, this occurs normally throughout life in all humans. But if it happens in a certain bad way, it can lead to the aggregation of that peptide with age in all primates. And so we've studied this, and actually cats and dogs at home, when they get beyond about 15 or 16 years of age, they may have these same amyloid deposits. So what happens in this neuron that has an amyloid parent protein is that there's an orderly cutting process by an enzyme called beta secretase, or BASE, and that releases part of this, and it leaves the rest of the protein still in the membrane or the cell wall. And then there's a second curious cut by an enzyme we call gamma secretase. Secretase means it secretes something, and that cuts in the membrane wall and it releases the amyloid beta protein. So what I've just shown you is what all of you are doing as we speak, and so am I. This happens normally throughout life, and everybody makes A beta. So do lower mammals, even animals that don't seem to get Alzheimer's. But when A beta teams up with another A beta molecule, 2 of them together form a dimer, one alone we call a monomer. They can even form trimers and tetramers and larger assemblies. And collectively, those teaming up of the A beta into assemblies, we refer to in science with the word oligomers, a few units or a few mers. So oligomers, we have come to believe through work in many labs, including Mark's work and my work and many others, are a type of smoking gun in Alzheimer's. They are noxious and they short circuit synapses. And those oligomers can make filaments or protofibrils and then fibrils, and those fibrils make up the amyloid plaques. So here I'm coming back to the image I showed you before. I've showed you how a normal protein called amyloid parent protein, or APP, can be cut by two enzymes, beta and gamma secretase, to release A beta. And then the A beta, if it stays as a monomer, is okay. But if it teams up with another monomer and makes a dimer, and then there are dimers of dimers, doublets and quadruplets, that's not good. And that, we think, can short circuit nerve cell function and lead ultimately to the other major lesion, the tangles of Alzheimer's, made up of the protein called tau. So A beta in the plaques, tau in the tangles, are something we desperately want to avoid allowing patients to accumulate. One idea about Alzheimer's, a hypothesis that's not yet proven, but there's more and more evidence for it, is that you could think of Alzheimer's as a syndrome that is not just one disease, but a disease with many causative factors that's caused by an imbalance between the production and the removal of this amyloid beta protein, the little black box in my last diagram, that leads to its gradual buildup in brain regions serving memory. And we believe that this imbalance can be caused by different genetic factors and or environmental factors. We don't know that much about exactly how environment leads to Alzheimer's per se, but there's some ideas about it. And one of them may be the experiencing head trauma can maybe trigger some of the lesions of Alzheimer's and even some Alzheimer-like symptoms. So this theory is called the amyloid hypothesis, and many people around the world have been working on it. And the drugs that are coming forward now are mostly directed to doing something about the problem that you see here that's outlined in this hypothesis. So the best evidence for this possibly being the mechanism behind Alzheimer's and therefore worthy of drugs that could modify it comes from familial Alzheimer's. For a long time, we've known that, like many other chronic diseases, Alzheimer's can run in families. And so the question is, how do defects in certain genes lead to Alzheimer's disease? And there's been a lot of progress in the last 20 years to understand genetic factors. And through the power of human genetics, scientists have discovered genes that lead to Alzheimer's. I'm going to show you an image here of many, many plaques in the gray matter, or the sort of the thinking part of the brain, the cortex of someone. And these many brown deposits are kinds of plaques. We call them earlier diffuse plaques or preamyloid plaques. And you see in the lower part of the screen, things are clear and white. That's the white matter of the brain. But in the upper part, most of the screen shows the gray matter of the brain, the cortex, and also the hippocampus, which is not shown here, accumulates many of these. And what's really shocking is for me to tell you this happened in a 12-year-old boy who died at Children's Hospital in Boston. And the question is, what would cause a 12-year-old boy to have these very much Alzheimer-like plaques of A beta or amyloid beta protein at age 12? And the answer is that this image comes from a 12-year-old boy with Down syndrome. And many of you have heard of Down syndrome and are familiar with it, also called trisomy 21, because there are 3 copies of chromosome number 21. It turns out that people with Down syndrome invariably develop this abnormality in the brain of Alzheimer plaques. And indeed, they get tangles as well a little bit after the plaques accumulate. And they eventually can lose ground. We know that Down syndrome patients have varying degrees of intellectual ability, and they have compromise of that, many of them, but they can do a lot. They can go to shelter workshops and they can go to school. However, when they get into their 20s, 30s, and beyond, they may lose ground. And that's because they seem to have acquired a secondary Alzheimer problem. Well, why is that? The reason is at the top of this list here. Chromosome number 21 is where the APP, or amyloid parent protein gene is, the gene that encodes the amyloid parent protein. And so Down syndrome patients have an extra copy of chromosome 21, and invariably they'll develop lots of amyloid plaques, A beta deposits, already in their teenage years. And so when this was known, scientists began looking carefully at the APP gene in many families with Alzheimer's and found some families that had mutations in APP. And in my lab and a couple of other labs, we studied that and we said that the effect on beta amyloid, the beta amyloid phenotype in the brain, is to increase the production of A beta peptides and especially A beta with 42 amino acids, A beta 42. Now, not just a year after these mutations were discovered, a gene variant called ApoE4 was linked to Alzheimer's quite dramatically. And it turns out that in this case, it's not increased production of A beta, but increased density of A beta plaques. That is, it seems to be mostly clearance of A beta that's faulty when you have a gene called ApoE4. And there are three variants of ApoE, E2, E3, and E4. Interestingly, E2 is protective against Alzheimer's, or at least amyloid deposition, and E4 is bad for it. About 20% of us in society will carry an ApoE4 gene that we got from mom or dad, either one. And that'll increase the density of A beta as we age. And then there's two quite rare genetic abnormalities, presenilin 1 and presenilin 2. The name sort of implies presenile Alzheimer's, and they get Alzheimer's disease in their 30s, 40s, and 50s. And those two similar genes, when they're mutant, can increase the production of this A beta 42 peptide or its levels. So this is the familial form of Alzheimer's disease. And in each case, when we found a genetic abnormality, you'll see an abnormality of A beta. And so that's support for the idea that A beta can drive it, because we have no doubt that these genes can trigger or cause the disease. So that comes to the end of what I'm going to share with you, just a couple of more slides, to remind you of this distinction between symptomatic treatments like donepezil, Aricept, and others, versus disease-modifying treatments, which we're working hard to try to get. We want to interfere with what you just heard about. We want to stop this buildup of A beta into oligomers and then to filaments and fibrils and plaques, and what we think is a secondary tau alteration that's very important. We think only when the patient develops tau tangles in nerve cells are they likely to begin to show serious memory symptoms. And so the drug that's furthest along, the treatment that's furthest along in trying to do something about what I've told you in this talk, is referred to by the name aducanumab. MAB at the end means monoclonal antibody, and aducanumab is a made-up name for a particular antibody developed by a company, Biogen, which happens to be headquartered here in Cambridge, Massachusetts. They did a trial, a phase 1 trial called the PRIME trial, and in that trial, they showed that over 1 year, they could lower the amount of amyloid, which is shown strikingly with an amyloid PET scan, tremendous breakthrough that was made at the University of Pittsburgh to find compounds that you could inject into the vein, cross the blood-brain barrier, and they would light up the amyloid plaques. And the pseudo-colored image, red and yellow, is what you don't want to have. These are excess amyloid plaques. Placebo for 1 year doesn't do much about this picture. These are actual images of patients in the trial. But giving 3 milligrams per kilogram body weight per month of this antibody, and you give it intravenously, begins to decrease the amount of A beta. And there's a dose-dependent increase, 6 and 10 milligrams, in this early small trial of 165 patients. So this isn't enough to get a drug approved, and Biogen went on to do large trials in 20 countries. Here's an example in a graph of one of their trials called EMERGE, and shows the longitudinal change from the baseline at the beginning of each patient's entry into the trial of their amyloid PET signal. So this amyloid PET scan, which is what I showed you here, the red and yellow signal is what we don't want to see. The decrease is striking and is dose dependent in this trial. And this was one of 2 trials. The other trial also showed lowering of amyloid in the brain over 78 weeks, although the other trial wasn't quite as strong, had a little bit less amyloid lowering. And then finally, what's the story on that? Does it matter to the patient? And the jury is sort of mixed in this trial. In purple is the degree of decline in a particular test. Um, Activities of daily living, a very important test that tells how people function in everyday life. The degree of decline is less here with the highest dose of aducanumab each month than it is with the placebo infusion given each month. And that difference is significant, but it's not a very small difference, and the patients still decline. So, what Mark and I and many colleagues in the field are trying to come to grips with is whether these data, which were impressive in this trial, but not near as impressive in the other trial, subsets of patients in the other trial showed some benefit, whether that will lead to an approval of this drug by the FDA. And that decision will come in June. So to summarize what I've told you, it turns out that Alzheimer's is a complex process that unfolds over years. And many of us believe that buildup of amyloid beta, followed by buildup of the tau protein, leads to the plaques and the tangles that Alzheimer originally described. We want to interfere with that. That is, we want to modify the disease. And we're hoping that drugs like aducanumab, which has not yet been decided upon by the FDA, will take us in that direction. There are a number of others that are very exciting that seem to do similar things. So with that, I'm going to stop and unshare my screen and ask if, let's see if I can unshare, stop share. Good, and I'll return to Mark, and maybe he'd like to take it from here. And then we're going to, of course, go into questions and answers after that.
Thank you, Dennis. That was wonderful. And thank you so much for that great introduction. I'm very excited to join and be able to share some information and thoughts related to Alzheimer's, and in particular, what we've been working on is asking if we can repurpose drugs that are already FDA approved to help with memory loss in Alzheimer's disease. And so I want to start by just redefining what is drug repurposing. And that is taking FDA approved drugs that are already on the market and asking, would they have any activity for different diseases than the ones that they were approved for? And so just to give you an example, there's been lots of work in drug repurposing for the COVID pandemic. And there's a drug called baricitinib that has it's an anti-inflammatory drug that's been approved for rheumatoid arthritis, but it's also now achieved emergency use authorization for COVID disease, and in particular to prevent patients that have COVID to progress to the severe form of disease where they have pneumonia, need intensive care unit, or and ultimately could pass away. And so this is an example where people were looking for ideas of drugs that already existed and maybe can help with a different disease type. And so I think because we're talking about diseases on the market, I just want to be very clear about two things. One is that I don't have any equity in any of the drugs that I'm going to be talking about, or in the companies that make the drugs that I am talking about. But also, just very importantly, what we're talking about here is research. And the drugs that I'm going to mention are, again, need further research. So this is what's called off-label use. And if we nominate a drug, that means it needs to go into a clinical trial for testing. It's not something that one should use to go to their doctor today and say, I heard this talk, could I go on this drug for Alzheimer's? This is still very much research, and I just want to make that very clear. And so this is a paper that just came out last week, so very excited to be able to tell you about it. We used a technique called machine learning, and I'll define what that is, to look in a more unbiased way at all the different drugs. We looked at a subset of drugs in a number of cases with Alzheimer's disease. And we kind of let the computer teach us what might be a signal that's worth following. And so what we started with was, as Dennis so nicely and beautifully laid out, is a set of brains from different places across the country. So in Chicago, in Jacksonville, Florida, and in New York. And these are brains that have come from autopsied samples. And the progression of the Alzheimer's pathology through the brains varies amongst these different brains. But you see the numbers here on the right. So we're looking at over 1,500 brains, and they will have different levels of progression. And then different parts of the brain were selected out, and then all the genes that were expressed in those regions of the brain were categorized. So now we have this enormous data set of 1,500 brains, 20,000 genes, and so it's too much data for a human to look at and try to decide is there a signal there or not. So that's why we decided to use computer approaches, and in particular machine learning. So what is machine learning? It's a term you probably hear a lot about, and it's something that I'm still learning about. But essentially what machine learning is it's taking a huge data set like that and asking the computer then to learn based on how we define it, and I'll tell you how we categorized it, to look for differences. And then in the second part, it builds a predictor, so that based on the data that you trained it on, so you taught it, then you build a predictor, and now you can feed in new information into the predictor, and then you get you get an outcome, a prediction coming out, and then you have to test that. And in Alzheimer's, what that means is we need to test it back in the laboratory, so either cell models, animal models, or ultimately to clinical trials. And because what we're talking about are FDA-approved drugs, then we have the opportunity then to move to clinical trials more quickly than drugs that come out that have not been in humans and we don't understand their safety program. So here's a complicated slide, but the heart of it is in the third, number 3. I'm not sure if you can see my pointer or not. I don't believe so, but I'll just talk you through it. So in the middle of the slide in 3, we took these brains and classified them into early stage disease, which is where people mostly don't have symptoms, medium stage disease, where they would have symptoms, and late stage disease, which would correlate more with moderate or severe forms of dementia. And then we asked the computer to tell us what's the difference between the early stage disease and middle stage disease. What's the difference between middle stage disease and late stage disease? How are the genes changing in these different regions of the brain? And from that, we built the predictor. And again, this is work of Artem Sokolov and Clemens Hug. And then that allowed us then to take other gene sets and feed it into the predictor and ask, would these particular genes be important for progression of Alzheimer's disease? And so we confirmed our predictor. We took known genes that are involved in cognitive decline, and that was the predictor worked well. It had a very significant signal. And so then we felt more confidence in our predictor. And so then we moved forward to ask about drugs, and FDA-approved drugs in particular. And so what we did is we took cells that grow in dishes in the lab. They're human neural cells, so they're brain cells. And then we treated those with drugs, these FDA-approved drugs, for a short period of time. And we looked at what genes changed in response to those drugs. And then we would take those genes that changed and feed it into the predictor and ask, if the drug would move genes in this direction, would it be helpful or not? Would it have a signal at all in terms of the genes that are changing with Alzheimer's? And these are the top hits that emerged. So on the left are drugs that are FDA approved. And on the right are drugs that are either have been in humans in clinical trials, or they're investigational drugs that are just getting into humans. So let's focus on the left-hand side. These are drugs that are already FDA approved. And we see a number of different drugs. On the left is the blue line. It's the approval. They've all been FDA approved. The first thing we did was ask, do any of these drugs cause toxicity? Because if they cause toxicity on the cells, well, that might be something that would be positive in our predictor, because neuronal death is part of Alzheimer's disease. And so one of the drugs dropped out. That you can see the red there. That one caused toxicity, so that one's off our list. But what emerged from this, which was quite exciting, is a class of drugs. The number one hit was ruxolitinib. But if you look down the list, there are a number of drugs that hit a protein called JAK, J-A-K. And JAK is a protein that's involved in inflammatory signaling in the body. And these drugs are approved for either rheumatoid arthritis or hematologic malignancies, so non-brain diseases. But including on the list is baricitinib, the one that I mentioned earlier, for that's been FDA had the EUA authorization for COVID. And so because of this class effect, we were particularly interested in that. And then secondly, and I won't have time to go into it, we've been working independently on a mechanistic study, and we've been seeing increased signaling, antiviral type signaling of immune cells in the brains of patients with Alzheimer's disease. And so this is just an example of this. This is a particular antiviral signal. This is antiviral signaling, an immune type of signaling that's in response to viruses. And we see that these are different genes that are increased in different regions of the brain that are relevant for symptoms in Alzheimer's disease. And in fact, this particular inflammatory pathway is blocked by those JAK inhibitors. And so we have then the computer led us down this road to this class of drugs. And then independently, we've had a mechanistic approach that has led to inflammatory signaling being present in patients with Alzheimer's disease. And in fact, to be more specific, what we see it is in about half the patients with Alzheimer's disease that we've been able to study in autopsy brains. And we have markers for this path, for this particular inflammatory signaling in the spinal fluid of living patients. And so we want to study whether, if we would change those inflammatory markers, would that have a positive effect on Alzheimer's disease? And I know later today you're going to be hearing about the cutting edge works from ALS, from Dr. Sudakovic, Dr. Gozala, it turns out we see the same inflammatory signaling in ALS as well as Alzheimer's disease. And so the clinical trial that we're about ready to launch in, we think, late spring or early summer, is what's called a basket trial, where we're going to take patients with Alzheimer's disease and ALS and look for their inflammatory markers in their spinal fluid. If we do see them in the spinal fluid, then they come into the trial and they'll be treated with one of these JAK inhibitors, baricitinib, for 24 weeks. So it's an open, everybody's going to get the drug in this clinical trial. And what we're really asking is does the inflammation in the blood, and also in the spinal fluid, is it reduced in the presence of this drug in particular? And we'll be also looking in the spinal fluid at markers of what Dennis was talking about, A beta levels, tau levels, and neuronal death markers as well. And so we're very excited about this. The trial is entirely philanthropically funded at the moment. We're also applying for grant support, but it's a wonderful expression of different foundations and grateful patients, et cetera, that have enabled us to take this drug candidate where we have convergent evidence and then move it into a clinical trial in a very short period of time. And so I just want to thank the team that I'm here to represent, a wonderful team. There's you can see that both here at Mass General, at Harvard, and then in London, And then particularly Artin Sokoloff, again, Clemens Hug did the computational work, the machine learning work that I talked about, and Steve Rodriguez did the work that was related to the in the culture models and things like that. So with that, I'm happy to turn it over and open it up to questions and look forward to all of your responses. Thank you for your attention and thank you for being here on a Saturday.
Great. Thank you very much, Mark. Right, and you can unshare and then they'll be able to see us. And I think the most important part of this after the presentations that Mark and I made in the search for therapy is to answer your questions. And we're prepared to do that. So I don't know how the questions are coming to us. Are they going to be articulated or coming by chat? Nothing in the chat room so far.
The questions are in order as previously discussed.
Okay,
So our
good. So
first
you want
question is, are dementia and Alzheimer's two separate conditions, or is one a precursor to the other?
So Mark, please go forward.
Yeah, so I think that when we Dennis did a beautiful job of laying that out. So just to very briefly summarize what Dennis had already said, and he's really a leader in this field, so you can't hear it from a better person. That dementia is a more broad term that talks about cognitive dysfunction leading to trouble with your ability to perform in everyday. And the most common cause of dementia is Alzheimer's disease by far. And so it's now patients can have changes in the brain for many years, as Dennis was saying, that are defined as Alzheimer's disease pathology. And those patients, many of them, and most of them, will, if allowed to live long enough, will develop the disease of dementia. And so you can have Alzheimer's changes in the brain of patients that are not having any cognitive symptoms. And so we would talk about those patients as having Alzheimer's disease pathology. But in terms of dementia, when people actually do have cognitive symptoms, the most common cause of that are changes that are due to Alzheimer's pathology. And Dennis, if you would want to say anything.
I think that says it all. I think for all of the patients and the caregivers who are listening in, this is what each family needs to deal with. What is the cause of my mother, my father, and my brother or sister not thinking as clearly? And specialists, neurologists, geriatric medicine specialists, psychiatrists are trained to figure out, is this dementia, number one? And if it is a dementia, what is the cause of the dementia? What is the form? And that's what we do in the clinic. So I think hopefully many of you who are dealing with the reality of Alzheimer's have gone past that point and have had a diagnosis by a specialist. So I think we can go to the next question.
Question 2 is this disease inherited, and how many generations does it affect?
So I'll take that one. And as I mentioned during my presentation, and these questions actually were formulated before we had chance to share our respective presentations. So indeed, we dealt with the notion of familial Alzheimer's. So this has been known for decades, probably centuries actually, to run in families. And yet there are any number of patients who come to see us in the clinic who don't have an obvious family history. So one doesn't have to have a family history. One doesn't have to have a clear-cut genetic pattern. But what I always quip is that one needs to choose one's parents very carefully. And time and again, both I and Mark and others that we speak with don't follow that advice. If you could, you would know that someone who has had development of cognitive failure in their 70s or 80s, that is a risk factor for developing it oneself. Now, let me hasten to add that by no means everyone who has a mother or father who developed Alzheimer's will necessarily get the disease itself. And actually, we have some evidence in society that the new attack rate of Alzheimer's, that is the incidence of the disease, may be declining a bit. perhaps due to better general health care, better nutrition, watching out for other things that cause brain problems like vascular disease. So we're not necessarily, we see a lot of Alzheimer's, there's no doubt about it, but it's not necessarily a true epidemic running away the way that COVID-19 is, for example. And so this disease has been with us for a long time. Genetics is a factor. There are multiple different genes. There are probably many dozens or hundreds of genes that can lead to it. I listed 4 in a table I showed you, and those are particularly noxious. And the one that would matter the most to people who are listening in today would be the gene we call ApoE4. So as I mentioned briefly, apolipoprotein E, ApoE, is a protein that's part of cholesterol particles or lipid particles throughout the body. And it was known for a long time that it was important for lipid transport. It turns out through careful research that ApoE4 is a major risk factor for getting Alzheimer's. Now, let me make one other point about genetics, and then we'll move to the next question. I wouldn't encourage people to run out and necessarily get tested for the genes that I listed or other genes linked to Alzheimer's. First of all, the only one that would make sense for the vast majority of listeners to get tested for would be ApoE, because that is by far the most common gene. The other genes I mentioned, so-called presenilin or the amyloid parent protein APP, those are exceedingly rare. And there the disease arises in the 30s, 40s, and 50s. So for all of us in society, ApoE would be the one we'd like to know about. And in some of the genetic testing services one can send DNA for, they'll let you know that you have an ApoE4, E3, or E2 gene. I don't advise that until there is a treatment. Once a treatment is approved, then we can do something about a genetic tendency, and then perhaps more people will come forward and be tested in an orderly fashion in clinical settings. But right now, I personally don't advise patients and families to get genetic testing. On the other hand, I do advise them that if there's a cognitive problem underway, they should see a specialist.
And the next question is there an age threshold for which you will likely not develop Alzheimer's?
Mark?
Yeah, so I would say that the age, chronological age, how much time you spent on this planet is the #1 predictor or risk factor for developing Alzheimer's disease, and that it continues as one ages, so that it grows in a what's called an exponential pattern. It's not even a linear or straight line. The curve is up, and so that as one ages, the risk increases. So that I would not say that there is a threshold at which if you reach a particular age, then you're home free. Unfortunately, it is something that correlates strongly with age at that stage. Now, that being said, I think as Dennis was hinting at or talking about directly was there's another concept out there of biological age where, it could be that by doing these changes in your watching your diet, exercising, keeping your mind active, engaging, and socializing, for example, those things could be something that people can do, loved ones can do to try to slow down the risk of developing symptomatic disease. And so I think that those things, and I believe the studies there's been some studies and there's further studies going on right now to look at this more carefully. But as far as the way I understand the data, I think that those things would be beneficial to people at all ages. And so I would encourage people to do those environmental activities to reduce the risk of developing these cognitive symptoms or progressing.
Okay, next question is there a relationship between poor sleep and development or progression of Alzheimer's?
Do you want to try that one also?
Sure, sure. No, so there's a it's a bit of a, I think it's a bit of a chicken and egg question. So there's, I think people that have poor sleep do have an increased risk of developing cognitive symptoms, including Alzheimer's disease. There's increasing evidence by labs across the country where the plaques, the A beta, that is the way that the brain clears A beta, seems to be pronounced during sleep, in particularly deep stages of sleep. And so if people have fragmented sleep, then their clearance of these toxins within the brain, including A beta, including tau, is probably reduced. And so that could lead to problems. On the other hand, it seems that patients that have pathology with Alzheimer's disease also, it can be a cause of fragmented sleep. And so it gets to be a challenge there. I would say that in general, sleep hygiene is something that I certainly counsel my patients on, is trying to adapt as good of habits as you can to preserve sleep as best you can. So things like don't watch, no blue lights, or don't watch screens an hour before you go to bed. Try to set a set time, whatever. It doesn't matter to me if you're a night owl or you're an early to bed, early to rise person. That's in part genetically entrained. So that I don't gonna say a specific bedtime, but I do think that there's, whatever works for you, kind of stick to a certain bedtime and try to pay attention to sleep, because I think that will help mitigate changes in cognition for a variety of reasons.
The next question is, are there any connections between diet and slowing the progression of Alzheimer's?
So that's a question that Mark and I and many other clinicians hear every time we see a family or a patient. And the answer is yes and no. Uh, the no part is that Alzheimer's is pretty much universal across the globe. And so people with very different diets will develop this disease. It's very prevalent in Asian countries, in Africa, and in Europe. So diet isn't a simple or direct factor that makes it more likely or less likely to get Alzheimer's disease. People with many different diets do get the disease, and the disease looks rather similar in those people, both clinically and pathologically. That is what we see in the brain when a patient dies. On the other hand, the yes part of the answer is that we increasingly believe that certain diets that people in the cardiology field mostly and in the nutritional field have told us are good for heart health, those diets may also be helpful in making it less likely that one would get Alzheimer's, or perhaps more correctly, slowing it down a bit and making it later in life that you might get it. So a heart healthy diet is sometimes also referred to as a Mediterranean diet. And many physicians believe, and the evidence is reasonably good, that people who emphasize the Mediterranean diet style with lots of green leafy vegetables and reds and yellow vegetables and nuts that grow on trees, but not nuts that grow on the ground, like peanuts. Those aren't the right ones. And a certain amount of modest wine consumption and oil, olive oil, those, and fish consumption, those are preferable for keeping a healthy heart. And that means less processed foods, less well-marbled meats. Doesn't mean one couldn't have any of those on occasion, but a weekly diet should emphasize more the qualities of the Mediterranean diet and somewhat less the classical American diet of the 1950s, which was lots of hamburgers and potatoes and things of that sort. So in that sense, diet is not a definite causative factor of the disease. I see patients with very different dietary habits and histories, and they all get Alzheimer's disease. On the other hand, it could be that if you do something about your diet when you're 40, 50, or 60, that could potentially lead to greater brain health, just as it leads apparently to greater heart health.
What age is the average age that someone is diagnosed?
So that is a great question. So the emphasis, I think, is trying to make the diagnosis as early as possible. So statistically, I think the numbers would be something in the average age is somewhere in the 70 to 75 range, roughly. I mean, but so much of that is dependent on when does the person or the family bring it to medical attention for the person to receive the diagnosis. And then also the level of comfort that physicians have to make the diagnosis. And so I was very encouraged, and Dennis probably played a role in this, but I was very encouraged that all physicians in Massachusetts this year have to get training in Alzheimer's, and that's coming down from the state. And so I know there was some strong advocacy for education amongst physicians for Alzheimer's disease. And I think that will help to bring attention to it, because not only does it, these types of things that we're talking about, changes in lifestyle and hopefully treatments emerging. I think in the history of medicine, we see that the earlier one treats a disease, usually the better the outcome. And I suspect that's going to be true in Alzheimer's. So the fact that we can be able to bring it to the attention of physicians earlier, and those types of mitigating strategies can be implemented, I think that will help. And Dennis even hinted at that it seems that the rate of Alzheimer's might be slightly going down, and maybe that's part of this what's the beginnings of what's happening. But I was very encouraged to see that there's going to be increasing education amongst physicians, and I think that will help them with the level of comfort of dealing with Alzheimer's, because I would completely agree. I wish we had enough specialists to see all of Alzheimer's, but it's possible, or it's not possible, to be honest, that every patient with Alzheimer's gets to see a specialist, at least right now. By helping primary care physicians be comfortable with it and getting people on the right track is going to be an important step to help mitigate the public health problem.
And I might add that about this issue of age, I agree completely with Mark's comments, and that the common age is in the mid 70s or mid to late 70s, but it keeps on happening in the 80s and in the 90s. I had a patient who was actually the wife of a professor at Harvard, where we teach and work, who brought his wife to me when she was 97. And he said that she wasn't doing the 1040 IRS form quite the way she used to. And of course, I couldn't possibly do the IRS 1040 form at any time in my life. She was able to do that. And she also played bridge very well, but she would renege or misbid. And sure enough, I said, well, your wife must be quite a remarkable woman at 97. She does all those things. But when I tested her, she was not as sharp as he led me to believe she had been. And she died eventually 4 or 5 years later at 102. And we found the abnormalities that I showed you in some of my slides of those amyloid plaques and neurofibrillary tangles widespread in the brain. So she handled it quite well. And I think there's no sort of maximum age at which you might get Alzheimer's. It is something that ramps up with life on the planet, time on the planet. It happens in other species, like all the primates we've studied, and in dogs and cats. And so I think the reality is that this is very age related. There's no maximal age. There's frankly hardly a minimal age. The youngest case I've ever heard of was 18 years old, which is unbelievable. But that was a young woman who had a presenilin mutation, a very aggressive presenilin mutation, which is one of those genes that, again, almost no one listening to us today is very likely to have such a gene mutation. They're quite rare. So, but mostly it's in the late 60s, the 70s, and 80s that we diagnose patients.
And our final question this morning is, can you discuss the MIT research about the use of gamma wave light and sound stimulation at 40 hertz as a possible treatment?
Yeah, so this is work that came from a distinguished neurobiologist, Liwei Tsai, at MIT. And she and her colleagues did remarkable experiments in animals, including mice that are engineered to have a mutation in this amyloid parent protein, the APP, that causes them to make too much A beta throughout life. And through a series of different, partially serendipitous experimental manipulations, they found that stimulating the visual system or the auditory system, virtually any sensory stimulus that was repetitive at a certain specific frequency of 40 times per minute, per second, that would lead then to some lowering of their amyloid plaque burden in the brain. So these mice were given a flashing light at a certain 40 cycles per second or 40 hertz stimulus, and that led remarkably to less amyloid over time. And they figured out that that seemed to be due to some activation of microglial cells, which are very important cells in the brain that help control inflammation and clearance of abnormal proteins. And so elevating microglial activity with a flashing light source or a flashing click or a repetitive click would actually be beneficial in mice. And that's gone into patients. Mark and I were discussing that before the session began, and we've each had a patient who's gone into a clinical trial by a small company, Cognito Therapeutics, I think they're called, where Cognito is offering a device that provides repetitive flashing light that patients use once a day or so. I don't know how exactly the trial is going now, but it's a very intriguing physical treatment of sorts that may work by stimulating amyloid clearing cells, referred to as microglia. I should say that that's just one example of a lot of different treatments. There are many dozens of clinical trials underway for Alzheimer's. And we should mention briefly that, for example, while we've focused a bit on the amyloid problem, the tau problem, the tangles of Alzheimer's are equally or maybe even more important as an abnormality that leads people to have trouble thinking. And so there are antibodies to the tau protein that are in clinical trials as we speak. There are remarkable new age techniques, type of genetic therapy where you can decrease the level of the gene that makes tau, so-called antisense oligonucleotides, and they might find good use in Alzheimer's. And there are trials underway to knock down the tau gene, not completely, but partially. People have even said that they might want to knock down that amyloid parent protein or APP gene somewhat. So these are new approaches. There's also various other kinds of stimulation approaches to the brain, magnetic stimulation, and others that people are testing to see if they might work for Alzheimer's. My own opinion is that the most likely thing will be the kind of thing that Mark displayed in his exciting new work, that is a small molecule, a pill of sorts, that will do something key to the abnormalities in the brain that will keep those abnormalities from causing a short circuiting of synapses. And also things like aducanumab and the other antibodies that are following it now in clinical trials. Those, I think, are the most likely thing to make a big dent in the Alzheimer process. That is, small molecule drugs and in infusion drugs like aducanumab, an antibody. And as Mark said, I also don't have any equity in these companies that make these drugs. It's important to mention. All right. Are there any other questions you'd like us to answer?
No, that's our final question. Just if you both have some final thoughts, we can wrap it up.
Good. So I think there's a few things we can still exchange. Mark, is there anything else you'd like to tell the audience about your experience managing the Alzheimer problem?
No, I think that I appreciate everyone coming out on a Saturday and to try to learn more about this important problem, and for all of your help and assistance and support, both with your patients, the loved ones, as well as trying to help move the science forward so that we can get to therapies. And in particular, as Dennis was emphasizing, the disease modifying therapies, so we can actually be slowing down the disease. And so we are, as researchers, I can say that we're very grateful for all of your support, and as physicians, we tip our caps to all the families that are dealing with it. It's an amazing. It takes an entire team to take care of one patient, and we are constantly inspired and amazed by all of the humanity and all of the care that we get to witness as trying to help solve this problem. So thank you for your time today.
And I'll add to that we're particularly grateful to the McCourt Foundation for sponsoring this public lecture for so many years. We've done this for many years in person, and now we're doing it virtually. And I think it's a very efficient way to do it, to reach a great many people. And they don't have to drive their cars to the local hotel where we give this talk. And we're very happy that Brian McCourt and his brothers and others in the McCourt Foundation have done so much to heighten the interest in not only Alzheimer's disease, but also multiple sclerosis. There'll be a session on that, and ALS now, and just be really concerned about driving brain research forward because it's such an enormous, important point. I'll also mention two other scientific points. One question I've always gotten over the years that doesn't seem to go away is whether Alzheimer's could be caused by aluminum or by some other metal ion. And we don't have evidence that that's a true causative factor. There's metal ions in everything that we contact and eat, the water we drink. And so metal ions are ubiquitous in our environment. And it's not out of the question that some metal ions could contribute a little bit to make things worse. But by and large, there's been very little evidence that metal ions, and particularly aluminum, which was brought forward when I first got into this field 3 decades or more ago, that's never really panned out, to use a bad pun. There hasn't been evidence that aluminum is an issue, but I know that that's still thing that people bring up to me. So it's not a matter of avoiding anything particular in the environment that would lead you not to get Alzheimer's. We just don't have evidence that specific environmental toxins or metals are truly causative of the disease. And then I'll also mention as a final point that we feel that patients and their families should be aware that there's no place in the world currently that has a bona fide proven treatment for Alzheimer's disease. So you don't need to flood the airways or get on the internet to try to figure out where's the doctor who has that Alzheimer's treatment that I can help my mother with or my father. That doesn't exist. You'll read in the papers what happens with the FDA and the European Medicines Agency, the EMA, and other agencies in terms of how they don't or do approve emerging treatments that have been in clinical trials. But there's no one out there, certainly including Mark and myself, who has a magic bullet for Alzheimer's. So I think it's very important to accept that reality and follow the news. The greatest source, I think, of information, and beyond even what the McCourt Foundation has helped with, is the Alzheimer's Association, which is in the United States and is around the world now. So the Alzheimer's Association has lots of information, and that's where you can find the latest word. And of course, you can see it in your newspapers and on when there is a treatment that anyone feels could be helpful for Alzheimer's, it will be front page news, and you won't have any trouble hearing about it. Right now, we really don't have such a treatment beyond the ones that are in clinical trials. So we do encourage people to enroll in clinical trials if they've seen a specialist and they're able to be directed to a clinical trial for Alzheimer's. And that's my final remark. And again, I want to thank Mark for joining me and thank the McCourt Foundation for sponsoring this. Thank you so much. Have a good day.
Thank you very much.